Hello and welcome to our video today. We'll be talking about the invasive species of the Asian longhorn beetle with Brooke Vickers, Carli Senese, and Charlie Carlton.
So the Asian longhorn beetle is right now devastating forests. It is causing oviposition cavities being chewed out. You can see on the left a picture of how big these cavities are and how these cavities are spread all throughout the tree trunk. Um, what they do is the larva is destroying the heartwood, and this creates holes in the bark, which usually are 3/8 of an inch in diameter. What this does is this destroys the quality of the wood, meaning it can be not used for production or manufacture. This also weakens the structural foundation of the trees, and it's causing economic cost with removing the trees. Usually, trees are having to be removed from parks, and this is causing a lot of cost. So right now, what's being done, the UC Riverside Center of Invasive Species Research, Department of Entomology, is introducing a natural predator, and they're also using quarantine methods and resistant tree species to protect against the Asian beetle. What we'd like to do is help integrate more effective quarantine methods by introducing a predatory species such as the entomopathogenic nematode into the beetle's habitat. So basically, our hypothesis is the best approach to stop the spread of Asian longhorn beetles, in our opinion, is to strengthen quarantine measures and introduce a predatory species, the entomopathogenic nematode, to the lumber. We think we can slow down the spread of the Asian beetle by instituting regular testing for this invasive species at hotspots for timber transit, such as ports and cargo ships. Outer inspections of timber may reveal the presence of the Asian beetle. The beetle causes holes on trees that are 3/8 of an inch in diameter. These cavities are usually quite noticeable. We believe by implementing this method, we can reduce the beetle's impact. Also, this approach aligns with physical reality because the entomopathogenic nematode have been shown to be used for biological control, and it's shown to be effective in the very well-known moth caterpillar, as well as crickets and grasshopper control.
So how we would collect our data for this experiment is over 2 months, we would track the number of holes in each tree in a controlled area that has been subjected to treatment. And to determine how many beetles and larvae are still alive, we would need to investigate and dissect about 5 trees per yield of lumber. The beetles would also have to be examined to check for the presence of the parasitic nematode. And our data would then be analyzed by using graphs that determine how long and how many Asian longhorn beetles and nematodes are collected over the 2-month period of the study. And our research can be reproduced by repeating this study in several regions over a 2 to 3 month period. During these studies, the climate will be diverse, and there even could be ecological challenges in place. Because our research can be reproduced, it is also falsifiable.
So with our hypothesis, we have 2 alternatives. The first one is the insecticides, which the main one is imidacloprid, and it can reduce the population of the Asian longhorn beetle when they are feeding on the leaves and the twigs of the treated trees, and eventually they die. This insecticide can be used in the seed, soil, or foliage to manage these invasive species. Along with the insecticides, we also have destroying infected trees. So when an affected tree occurs, there's a removal process that happens, and after they remove the tree, they either chip it like in the photo, or it's burned in a quarantine area. And with these hypotheses and with our experiment, we have personal bias. So there's 2 types that we came up with. First one is insecticides should be used because it is the most effective way to manage or remove the Asian longhorn beetle. The second one is we could use local indigenous knowledge to conduct our research and data collection. And for diversity, we're using the alternative hypotheses to create new experiments to help manage the Asian longhorn beetle, like testing insecticides in different regions and to see which one is the most efficient. So instead of using imidacloprid, we can use another insecticide to see if it's better or less than the imidacloprid. Another test we could do with these hypotheses is compare the most efficient or effective insecticide in a controlled quarantine environment or in the field where there's no quarantine at all and see which one is the most effective. Thank you everyone for listening to our presentation. Have a good day.